Okay, welcome to Frontiers in Brain Health. I'm Dan Krosik, the Deputy Director here, and we are honored to have a really exceptional speaker and an exceptional topic. When we think about health and brain health, I would say there's no argument that exercise is going to be part of that. The question is how, and the question is really what does it do for us? And so that's going to be our theme today. We are recording, so this is going to be available later, and we do have virtual audience as well as welcome to the large in-person audience. We will get to questions from both audiences, so use the Q&A function if you are joining us virtually for this talk. And today we are fortunate to have Dr. Kirk Erickson with us. He is really one of the world's experts in exercise and what it does for us, particularly above the neck. So we often think of exercise being, of course, helping our cardiovascular health and our muscular health, but it also affects our brain health tremendously. It's a whole big living system, as we know. So Dr. Erickson was trained at the University of Illinois, Urbana-Champaign. He was also a postdoc at the Beckman Institute there. He was faculty at the University of Pittsburgh for a number of years before taking on the role as the Director of Translational Research and the Blair Endowed Chair at Advent Health, the Advent Health Research Institute, where he is currently. And he really has a vast program of research that focuses on those relationships between physical activity, exercise, and brain health, and does so across the lifespan. So many age ranges will all benefit from this. He's published over 300 articles, giant in the field. So we are so thrilled and honored to have Dr. Kirk Erickson. Welcome, Kirk.
Thank you so much. You can hear me? Yep, good. Thank you so much for the introduction, and I'm going to talk to you today about the importance of exercise for your brain. If you were at my talk yesterday, there's going to be a little bit of overlap, but I also try to have this a bit separate and a little bit more data in this presentation than I presented yesterday, which was a bit briefer. Whenever we talk about exercise in the brain, we always have to start talking about cognitive aging, especially. Most of the work on exercise in the brain actually is really in the context of late adulthood. There's a growing body of work in children. So that's kind of the other smaller mountain of data that exists is the benefits of exercise on childhood brain function. And then we've got a bit of a gap, actually. Adolescence, we don't really know that much. Young adulthood, we don't really know that much. Midlife, we're learning a bit more. But especially on cognitive performance. When we look at mood and other outcomes, it changes a bit. But I'm going to focus most of my presentation here on the effects in late adulthood. Now, I probably don't have to remind most of you in the room about the age-related cognitive changes that occur across numerous cognitive domains. We see relative stability in cognitive performance, maybe up until around the age of 60. Some argue younger than that is when we start to see some cognitive changes, but across most or many different cognitive domains, we see some gradual decline across the late adult age range. Now, what happens to the brain is that before we can even detect some of these cognitive changes, the brain itself is changing. And so, for an example, this region here, the dorsolateral prefrontal cortex, we know that this area starts to shrink as we get older. In fact, right around the age of 30 is when we start seeing some decline in the size of this region. This is a plot of about 200 people between 18 and 80 years of age. And you can see the decline in the size of this structure over that time frame. This structure right here is the hippocampus. So the hippocampus is sitting right in the medial temporal lobe, and it's involved in supporting memory function, especially episodic memory function. It also declines in size with age. So as you can see, it's a slightly different function where there's relative stability in the size of the structure up until right around the age of 50, and then it starts to decline in size after the age of 50. The rate of deterioration of this structure is actually predictive of onset of memory problems and of mild cognitive impairment and Alzheimer's disease-related impairments. So we see these changes happening in the structure, the morphology of the brain. What I want to point out here to you, however, is not the average line, which has been shown across many, many studies at this point. Instead, I want you to focus on the variability around that curve, on each of these curves. So let's take this person right here. This person is about a 70-year-old individual, but if you look at the size of the prefrontal cortex, they actually have the size of the prefrontal cortex equivalent to somebody much, much younger, about 25 years of age. So if I'm 70, I want to have a brain like this, right? This person who's about the same age actually has the size of the prefrontal cortex equivalent to somebody about 25 years older, almost 100 years old. So this is a significant amount of individual variability in the size and shape of these structures. And we see the same type of variability across all these other structures, including the hippocampus. So this brings up a very important question that was raised several decades ago now by our group and others. And that is, what factors contribute to this type of variability? Are those people, this person right here and that person right there, are they just genetically lucky? Or have they done something different throughout their lifespan that's allowed them to age more successfully, or at least their brain to age more successfully? And if we can identify what those factors are, maybe we can intervene. We can intervene to slow down or alter the trajectory of change. So this is really where exercise emerges onto the scene as a possible way of not only preventing decline, but also influencing and actually improving and altering the rate of decline. We've known for many years that the benefits of exercise on various cardiovascular, muscular, cardiometabolic, metabolic outcomes, risks for mortality, risk for cancers. But it's only been actually fairly recently, over the last several decades, that the brain has now been a real source of interest by scientists to examine the impact of exercise on this organ. This is partly due to recent neuroimaging techniques, right? So many decades ago, even a few decades ago, we didn't have the capability of really examining the brain, the living brain in humans the way that we have, the way that we can now. We relied mostly on interventions that were looking at cognitive outcomes, or there was animal research, and I'm going to talk a little bit about some animal studies in a little bit. We also have epidemiological studies, observational studies in which we can follow people for many years, and so there's those studies as well that examine physical activity earlier in the lifespan and examine the risk for experiencing dementia or some sort of neurodegenerative condition later on in life. And a lot of this work has now been described and discussed in numerous publications. This is a paper that I'm putting up here, recently published, where we talk specifically about the promise of physical activity and thinking about cognitive aging. Instead of thinking about it as this deterioration and downward trajectory, we're thinking about it as in a way that is more promising and more of an optimistic approach, that we can modify the trajectory. But when we look at across the data, there's a lot of consistency. There's a lot of consistent patterns. This is a really interesting meta-analysis published in 2011 from observational studies. So this is not randomized clinical trials, observational studies. Physical activity assessed earlier in the lifespan was associated with a reduced risk of experiencing cognitive decline later on in life. In this particular meta-analysis, there's about 33,000 individuals across all of these studies. And this general pattern has been replicated across many different meta-analyses. So we know that physical inactivity is a risk factor for many different neurodegenerative conditions. including just general cognitive decline, as well as Alzheimer's disease. So if we pull out those studies that just focused on risk for Alzheimer's disease in particular, here too, we see all of these studies on this side of the line associated with a reduced risk of developing Alzheimer's disease. In addition, there have been meta-analyses of randomized clinical trials. This is one of the more important meta-analyses published in 2003. So quite a few years ago, and this study included randomized clinical trials that went back into the 1980s. And this is something that is often overlooked, is that randomized clinical trials of exercise, looking at cognitive outcomes, goes back to at least the early 1980s. This has been going on for a long time, actually. And what this meta-analysis found, there's now been, I believe, 24 meta-analyses of this literature, and 22 of the 24 all show the same pattern. So what they show is that engaging in exercise, randomized clinical trials of exercise, show improvements across most cognitive domains. There's two really important take-home messages of this particular slide. One is that you see exercise has a fairly broad effect across all cognitive domains, meaning that there's a fairly general effect of exercise benefiting cognitive performance across most domains. However, you see that there's this one bar over on the left-hand side that's capturing executive functions. For some reason, executive functions seem to be more sensitive to exercise. You might ask why, and that's a very good question. We still don't have a very good answer for it, but I'm hoping to address that a little bit later in my presentation. So, we have observational studies showing that if you engage in greater amounts of physical activity earlier in the lifespan, you show a reduced risk of developing numerous neurological conditions later in life, including Alzheimer's disease. And when we bring older people into the lab, get them exercising for several months, we show improvements in cognitive performance. So this was all literature. This was published in 2003. This was all science and meta-analysis and literature that was published really before we had a lot of our neuroimaging techniques that allowed us to peek inside the skulls of these individuals to see what's happening to their brains. So one of the first studies that we did, we wanted to examine associations between a marker, a measurement, called cardiorespiratory fitness. So when we talk about fitness, we can actually talk about it in numerous ways. Flexibility and balance are types of fitness. Muscle strength is a type of fitness, but there's also lung capacity, your aerobic capacity, essentially. So this is a measure. What we do is take measures of cardiorespiratory fitness. And in this particular study, we brought in 165 adults between 59 and 81 years of age. We we tested them on a treadmill to measure cardiorespiratory fitness. We put them into an MRI, and then we assessed the volume of their hippocampus. Now, remember, going back to that figure that I showed you just a couple of minutes ago with that variability around the curve for the hippocampal volume. Our question was really, could cardiorespiratory fitness explain some of that individual variation? So we took the MRIs, we applied a segmentation algorithm, we segmented the hippocampus from the structures around it, and this is what we found. We found that higher levels of cardiorespiratory fitness were associated with larger hippocampal volumes. This effect was significant, even when controlling for variation associated with age, sex differences, years of education, and intracranial volume. So what's important is that these people up here don't just have bigger heads. Okay? So we adjusted for that. So this is very interesting. This is very provocative. It suggests that a physiological measure of fitness is related and explains individual variation in hippocampal volume at a time period of life where we're typically seen decline in the size of this structure, and decline in this structure is related to onset of Alzheimer's disease. So very interesting, very provocative. What's the problem with this? The problem is that it's a correlation. It's just an association. So if you think back to your statistics courses, we can say that we cannot make causal claims from a correlation. We cannot claim from this association that increasing cardiorespiratory fitness would actually alter hippocampal volume. We cannot make that claim here. Nonetheless, this is very interesting, very provocative, and this particular finding has now been replicated across numerous other studies and age ranges as well. So this is very interesting, but it suggested to us that we needed to do an intervention. If we do a randomized clinical trial in which we experimentally manipulate exercise behaviors, show changes in cardiorespiratory fitness, can we actually modify the size of the hippocampus? So we bring in older, relatively sedentary adults into the laboratory. We do baseline assessments. We randomize them to one of two conditions. The two conditions are really important. So we have either a brisk walking group or more of a stretching group that comes in and does light intensity stretching. They both come in for the same amount of time. So they're getting the same amount of social engagement. They're getting the same amount of health instruction. They come in for the same number of days per week. So the main differences here are the intensity and type of physical activity. Where the brisk walking group, we make sure that they're exercising at a moderate intensity level. And we do that for 6 months or 1 year, and then we do all of the follow-up assessments that we did at baseline. So this paper was published in 2011, and we reliably improved fitness levels in our walking exercise group. So our walking intervention was successful based on this measure. What happened to the brain? And I'm going to focus here on the hippocampus. So up on top, you see the hippocampus, and with the yellow on the top panel, and you see the left hippocampus and the right hippocampus. In the red line is our control group, and they're showing a slight decline in the size of this structure. This is a natural aging process. Remember, I told you that annually, there's about a 1.5% change in the size of the hippocampus. So every year that you get older, about a 1.5% decline in the size of the structure. So the red line is consistent with normal aging. The blue line, however, showed a 2% increase in the size of the hippocampus. So there's a couple of really important messages that came from this paper. One is that the older adult brain retains its capacity for plasticity. And only 12 months of moderate intensity exercise was able to modify the size of the structure that typically deteriorates in late adulthood and that is predictive of onset of memory problems and Alzheimer's disease. It's pretty remarkable, especially when you consider all of the pharmaceutical interventions that have been trying to do the same thing for many, many years. So this was a really important finding. Since this finding, there's been some studies that have replicated it and other studies that have failed to replicate it. So there's been a number of meta-analyses. We completed this meta-analysis just a few years ago, and one of the things that I want to point out here is we decided on this term preserve, because what is very clear from this literature is that at the very least, exercise is a good way of preserving the size of the hippocampus, even if we're wrong and the prior results are wrong and that we can't increase the size of the hippocampus. We are pretty confident now that it's good at preserving the hippocampus over the adult lifespan. So this is really a very important outcome and continued replication of this work. So you might be wondering, well, what's happening to memory? Because the hippocampus is really involved in memory formation. So what's happening to memory? So there have been a lot of studies, randomized clinical trials, examining the impact of exercise on episodic memory performance in late adulthood. So we examined this really recently in a meta-analysis. We pooled all of the studies that we could find in this area and examined what does exercise do. And this is a very, very interesting story and tells us a lot about why some studies actually fail to find effects on episodic memory. So here's what we found. You can see all of the studies. There's actually a lot of exercise, randomized clinical trials of exercise, all in late adulthood, all examining the impact on memory performance. Okay, so that's something really remarkable here. It shows you the growth of this literature and the interest in this area. Look down this column here, where it shows p-value. Do you see many that are significant, that are less than p less than 0.05? Not many. Meaning a lot of individual studies do not show significant effects. However, when you pool across all of these studies, you do see a significant effect. So, why is this? The problem has been that based on this effect size, so this is a significant effect, based on this effect size and based on the variability, all of these studies have sample sizes that are way too small. So you only really can detect the effect when you pool across all of these studies. So based on this effect size, we found that we require about 202 people per group to be sufficiently powered to change episodic memory performance. Most studies had about 30 or 40 people. So there is an effect size of 0.28, small to moderate effect on episodic memory, but it is there. So volume we're seeing, and we're seeing some patterns on episodic memory performance. So with that, you might be asking, and I'm hoping you're asking, what are the mechanisms? How does this actually work? Well, we've tried to summarize several different ways of conceptualizing mechanisms. And here I've shown you, this is a figure from the paper published in 2022. And what you see is that we've divided this into 3 different levels of mechanisms. Quite often, many of us, when we start thinking about mechanisms, we jump right away to what are the cellular and molecular pathways. And certainly that's one of them, but we can also ask the important question of level 2, we see these neuroimaging changes, are they actually mediating the cognitive and behavioral outcomes? So let me pose it in a slightly different way. I showed you earlier volumetric changes in the hippocampus. A skeptical person might ask, well, could this be just a meaningless byproduct of exercising? That it doesn't have any relevance to memory or behavioral outcomes? And that's a perfectly valid skeptical question to be asking. So what we need to do is actually test its relevance and its association with cognitive changes or any other behavioral changes. So in this pathway, these are not mutually exclusive pathways, by the way, right? So anything that's happening in this level 2 must have some sort of cellular or molecular changes being induced that influence those volumetric changes. But on level 3 is a level that is often ignored. And let me frame it this way, and I framed it this way last night as well to the audience. Exercise is a great way to get people to sleep better. So maybe sleep is the pathway. Maybe sleep is the key. If we modify sleep, then we have better memory function. So exercise is improving memory function because it's improving sleep. Sleep would then be the mediator, sleep would be the mechanism. Okay? So we can think about mechanisms on multiple levels. We don't have to just think about it on the reductionist cellular molecular level. We unfortunately have a lot more data on that side. We unfortunately don't have a whole lot of data on these level 2 and level 3. So animal studies, we can do a lot with animal studies, but there's limitations. One of the benefits is to better understand cellular and molecular pathways. We can harvest brain tissue. Can't do that very easily in humans. We can experimentally block pathways to really derive much stronger conclusions about causality. We have greater genetic and environmental control, but the translation to humans is always going to be a question. In contrast, human studies, we don't have concerns about translatability. We can do randomized clinical trials. We can assess brain biomarkers. We can also assess peripheral physiology to look at other markers, and we can attempt to use statistical mediation frameworks to better understand causal pathways. So let's focus on what has the animal literature shown us. The animal literature has actually shown us that exercise is a great way of producing new neurons. Where? In the hippocampus. Interesting. The same region that I was showing volumetric changes. So exercise doesn't impact neurogenesis in the olfactory bulb, but it does do so in the dentate gyrus of the hippocampus. So there's some regional specificity here. Interestingly, exercise not only induces new neurons, but it also induces new capillary beds, new vascular beds, through numerous regions: the cerebellum, motor cortex, the hippocampus, basal ganglia, most regions, a lot of regions that have been assessed so far. This is a paper going back to 1990, actually showing increased capillary beds in the cerebellum after exercise. But what's really interesting, what I find really interesting about this work, and a lot of the animal work in this area, is that what we've been learning is that changes in the capillary beds are actually a precursor to changes and really a requirement for changes happening in neurogenesis. So if you block angiogenesis from occurring, you can block the neurogenesis, the exercise-induced neurogenesis. So this is very interesting work, and suggests one thing has to lead to another. We have to put the whole pathway together. And this is from animal studies, but and this is one more molecule I forgot. Brain-derived neurotrophic factor, BDNF. This is another important molecule. Going back to 1995, we know that exercise increases the production of the expression of this particular molecule. Where? Primarily in the hippocampus. Keeps coming back, right? The hippocampus. Now, BDNF is a very interesting molecule. It is released from the synapse into the synaptic cleft, and it binds with a high affinity to this receptor called TrkB. And BDNF, once it binds, it induces a whole cascade of events inside the cell, and so it induces synaptic plasticity, dendritic expansion, and it's associated with learning and memory. So we can experimentally block in animals, in non-human animals, we can experimentally block this particular receptor. And what happens? We block learning and memory. So this is really critically important in learning and memory function. And I just showed you exercise induces increased BDNF levels. So, and I'll probably skip over this really quickly, but obviously there's other pathways between the skeletal muscle going to circulating factors then inside the brain that influences proliferation of new neurons and capillary beds. Going back to this figure, however, we collected blood on our participants and we measured BDNF, because we can measure BDNF in the periphery of humans. And what did we find? We found that changes in BDNF levels over the course of the 12-month intervention were correlated with changes in hippocampal volume. This is very, very interesting. What we've now just discovered is a blood biomarker that's related to the changes in hippocampal volume. Very, very intriguing. Now, again, I want to take a step back. Let's think this through. This is a correlation. Again, not a manipulation of BDNF. This is a correlation. Nonetheless, it's very interesting because it aligns very nicely with all of the non-human rodent work that I just showed you. So maybe we're closer to rats than what we'd like to think we are. We also see changes in brain connectivity metrics, and in turn, changes in brain connectivity are also correlated with BDNF as well as a few other markers. You might ask, well, what about amyloid or tau or any marker, Alzheimer's disease marker of pathology? This is a very interesting question, and Sandy and I mentioned last night at my presentation, Sandy and I have had some conversations about this. I've got some ideas about what's going on here, but in this one study published by our group in 2017, we analyzed data from 149 people that were part of an observational longitudinal study, and what we found was that physical activity was associated with a reduced risk of cognitive impairment 13 years later. This is consistent with a lot of other research. But then what we found was that this 9-year measurement of amyloid was actually statistically mediating this association, suggesting that physical activity-induced changes in amyloid might be mediating the cognitive risks for cognitive impairment. So how accurate is this? Unfortunately, it doesn't seem to really hold up. We recently conducted the systematic review and meta-analysis on physical activity in amyloid beta in middle-aged and older adults, and this literature is really a mess. I have no confidence at all that physical activity is actually associated with any reduction in Alzheimer's disease pathology. Okay? So this relates back to a comment that I made last night, that I don't think physical activity is reducing risk for Alzheimer's disease because it's influencing Alzheimer's disease pathology. I think it's acting through other pathways. So we have other moderating factors that we could consider. So for an example, you go out for a run, but every time you go out for a run, you come back and have a greasy burger. That might be mitigating some of the impact of exercise on cognitive and brain outcomes. Whereas if you go out for a run and then come back and get into an intellectually stimulating atmosphere, maybe that's augmenting. So we're starting to understand some of these moderating factors. And one of them appears to be APOE genotype, so the genetic risk factor for Alzheimer's disease. Now here too, we still have a lot to learn about how these pathways come together and whether APOE is really moderating much here. But here what you see is people who have low amounts of physical activity versus people that have high amounts. And these are people that do not carry the genetic risk allele versus people who do carry the genetic risk allele. The bar that jumps off the screen at you is this one, right? Low amounts of physical activity and carrying the genetic risk allele. They have high amounts of amyloid in their brain. But what I'm most interested in are these guys right here. These guys have a genetic risk for high amounts of amyloid. What the main difference is that they're getting high amounts of physical activity. High amounts of physical activity seems to be completely mitigating the genetic risk for having high amyloid. So this is very interesting and suggests that maybe some of our lifestyles might be able to offset some genetic risks that we might be having. This needs to be replicated in larger samples. It needs to be conducted in an intervention type of format, randomized clinical trial, and so on. I've got numerous studies going on. I can't get into all of them here, but I do want to focus for just the last few minutes here on investigating gains in neurocognition and intervention trial of exercise, IGNITE. This was a very large NIH-funded project, and it was really designed to be the largest and most comprehensive phase 3 dose response randomized clinical trial of aerobic exercise, specifically designed to examine neurocognitive outcomes in late adulthood. I'm going to briefly show you a result here. We actually have a lot of very interesting results. Too much to really share with you here, but I'm going to focus on one. This is not yet published. It's submitted for publication. We analyzed the baseline data. Notice that we had 648 people in this study. So this was a large sample, large study. We had 3 sites, University of Pittsburgh, University of Kansas, and Northeastern University. You can see APOE4 carried, so about 27% of our sample were carrying the E4 allele. And we measured VO2, so we measured cardiorespiratory fitness. Remember the same measurement I showed you earlier, that cardiorespiratory fitness is associated with hippocampal volume. We measured that here as well, but we also wanted to pull this measurement apart because, and you can see here, what we did was we examined all of the components of VO2. to examine whether there's some aspects of physiological function within the VO2 measurement that contributes or explains some of the associations that I'm going to show you here in a minute. And so this is kind of a description of the sample. We conducted a very comprehensive cognitive battery lasting 4 to 5 hours on all 648 participants. And we then conducted a factor analysis, confirmatory factor analysis, with a good model fit. And we have these 5 cognitive domains: attentional control, working memory, episodic memory, processing speed, and visual spatial function. And what we found was that higher VO2 peak, higher fitness levels, were associated with better performance in every single one of these cognitive domains. The effect size is actually about equivalent to years of education. You know how we always use years of education in all of our statistical models examining cognitive performance? And there's plenty of reasons to do so. What's interesting to me is that cardiorespiratory fitness actually explained about the same amount of variance in cognitive performance. But cardiorespiratory fitness is never measured across all of these aging studies. So we see this association. This is very important initial finding. And we broke it down by male and female, and you can see that females tend to show a greater, slightly stronger association than males. Here is another finding that I'm very interested in, is the middle one: years of education. Years of education is also known to be protective. More years of education, more protected you are. What we're finding here is that years of education is associated with cognitive performance only at low VO2 levels, low fitness levels. If we look at people who have few years of education but remain high fit, there's no more differences in the years of education. Really, really interesting. In addition, one thing I'm not showing you here is I looked at an interaction with APOE. No interaction with APOE, which means that carriers and non-carriers show the same benefits of having higher fitness. So we are now doing a longitudinal analysis of this, this is the IGNITE study, where we did this for 12 months. We did all post-intervention assessments. So yes, we have now moved from IGNITE to FLAME, which I'm very excited about. And we had no contact with our participants for 5 years, and now we're funded to do this 5-year follow-up of all 648 people, doing fitness, DEXA, pulse wave velocity, blood markers of Alzheimer's disease pathology, BDNF. We're getting everything. This has been translated to public health policies this was the Physical Activity Guidelines for Americans, published in 2018. So I'm very excited. I don't want to just keep these studies and this research in the laboratory setting. I want to make sure that people understand the importance and impact that physical activity can have on enhancing and maintaining brain function throughout the lifespan. And by the way, this 2018 guidelines were reviewed and published in 2019, as you see here. So in conclusion, exercise has widespread effects on the brain. That being said, there seems to be some regional specificity. Notice I kept bringing up the hippocampus. If you look across the literature, the hippocampus keeps coming up in these studies over and over and over again. The frontal cortex and anterior cingulate cortex, I think, are the other areas that keep coming up in this literature. Why is there regional specificity? We don't really have a good grasp or good understanding of that just yet. We still have a poor understanding of the mechanisms. We're starting to get there with larger sample sizes, harmonization across groups that are doing these studies, so we can pool data, pool effect sizes, and better understand pathways. Understanding heterogeneity of response and individual differences remains a major gap. One of the problems and one of the reasons why it is still a gap is that you need large sample sizes to actually better understand individual differences. In most studies, they don't have the sample sizes to really examine individual differences. IGNITE was designed to try to address some of those individual differences. And exercise may have long-term health consequences for many diseases that affect the brain. Not just diseases of the brain, diseases that affect the brain. So I just finished a study in which we targeted women with breast cancer, brought them in, gave them exercise for 6 months, and we saw significant cognitive improvements in the sample, which we've shown previously typically show some cognitive deficits with their therapy. So exercise does have the potential for really enhancing the brain. So with that, I just want to thank all of you for listening here and my entire team for all of the work that they do on all of these studies and funding and collaborators on all of these projects. So thank you very much.
Fantastic information, and so painstaking to cover all those different areas. So we do have time for questions. We'll go ahead and open it up. And here we go. Yeah.
Hey, Kirk.
Hey.
It's really interesting to me, and I came from a different world, but why does the pharmaceutical mafia spend so much time on trying to find that pill when you're really showing that you're making a difference with behavioral changes? And first part of my question, I'm going to cheat and have a second part. How valuable do you consider you being part of these meta-analyses in terms of kind of bundling what's happening in these smaller trials, and how valid are those types of findings?
Yeah, two good questions. The answer to the first one, I would say money. You can't patent physical activity, and it's more difficult to make money out of it from a industry perspective. So I think that's an issue. Now, I've actually been approaching several pharmaceutical companies and telling them two things. One is that they are not even measuring physical activity. So at the very least, that's an untested, unexplained source of variance in their own data. The second thing is that I am guessing that there are interactions between pharmaceuticals and physical activity. So that's something that I think they should be interested in. And I've even taken it on a third route, saying that at the very least, you might want to know, is it safe to take your medication while people are exercising? So even from just a general safety perspective, it would be useful to say, you know what, you can still continue exercising safely while you're taking this medication. So I've been approaching them, and I've had a number of conversations like this, but nothing yet. So can say that. The second question is around really the size, the conduct of these studies. There's a couple of things that I really want to be cautious about. Large studies take a lot of resources and a lot of money. And a lot of groups around the world just don't have that. So I don't want to be sounding like I want to be limiting the science just because it's not going to be having the sample sizes or the types of assessments that I think we generally need at this point. Because I do think that smaller studies can be quite informative, especially if you have some of the right assessments. So I've been going around, I travel around the world actually meeting with different groups, sharing with them some of the results that I find, some even unpublished results, saying, look what we're seeing with this measure, here's how we did it. And so as you're designing your study, maybe you want to consider doing it this way. Or here's the intensity level where we're starting to see some shift. So maybe target this intensity level. So I'm trying to provide some guidance, and again, providing some harmonization around these studies around the world, because right now I think it's contributing to some of the muddiness in the literature.
I'm next. No, we'll let our board member go ahead. Go ahead.
Yeah. So the question is around dose response. And dose response is a very interesting question when it comes to exercise, because you can think about dose in a number of different ways, right? Quite a few. The intensity of the activity. There's low intensity or light intensity, there's moderate, and there's more vigorous. You can think about it in terms of the frequency of how often, right? How many minutes a week do you get this? And does it occur in 30-minute chunks, right? Are you accumulating this? How are you accumulating your 150 minutes per week, if that's what you're targeting? There's also the type, right? So the mode of exercise. Are you doing resistance training, or are you doing more aerobic types of training? And there's other factors that play a role in this as well. Recently, we've been starting to talk about the density of activity within a 1-week cycle. So for an example, you might be able to think about the weekend warrior, right, where they're putting all of their 150 or 300 minutes in one day almost, right? So what impact does that have compared to balancing it out or distributing those 150 or 300 minutes across the week? So there's a lot of very interesting questions around this. And the basic answer, I'd have to say, is right now we don't have a good answer. So the answer is we don't have a good answer. Ignite was actually designed to look at dose response. So what we did was we divided the group. I didn't show you here, but we divided the groups into 3, one of which received light intensity activity for 150 minutes per week, one that received moderate intensity activity for 150 minutes per week. So we made the amount of minutes per week equivalent and just manipulated the intensity. Then we have a third group where we kept moderate intensity equivalent, but we gave them 225 minutes per week. So with that, we're going to be able to test and compare the differences in intensity versus the differences in the minutes per week achieved or accumulated. So this study, as well as a few others that are going on right now, will hopefully start to address some of that dose response question. What you're getting at is something that I really focused on last night as well, in my presentation last night, is prescription. We need to be able to prescribe exercise, and we need to be able to say, for the brain, for the sake of the brain, and we need to say how much is needed. How much should you be getting? And really, what a lot of people are asking me when they ask that question is what is the minimal amount of activity they need to get in order to achieve some brain outcome? And unfortunately, we don't really have a good answer for it.
Can you say what you did, for example, in this study where people did show an improvement?
Yes, and that's what a lot of studies have been doing right now. So we know intensity, for an example, is very important. But those studies, a lot of them have been observational, there's flaws in them, so we really needed a randomized clinical trial that manipulated dose in this way to be really confident. And that's what we try to do in Ignite. Yep. In the back.
If sample groups and money are prohibitive factors in collecting information that you need, would you consider using AI as a tool to get the information you like?
Yeah, there's a lot of basic answer, yes, but it's not going to give you the randomized clinical trials necessarily, right? You still need to be able to conduct those in a way that where you have that type of control. But getting large sample sizes and getting data collected on the population, I think AI is going to be a fantastic resource, a fantastic way to go. The challenge is these randomized clinical trials. They take a long time. There's a lot of regulatory paperwork and other things that have to go into them, and it takes a lot of resources, facilities, people, and when I travel around, a lot of times people just don't have those resources. So I want to be cautious when I'm talking about we need more studies like this, because realistically it's not going to happen. So I do want to be cautious about that. But I do think that the efforts that I've been trying to do in harmonizing, communicating the science, communicating what we're finding, even before we publish it, like I showed you here, I showed you some preliminary data not yet published. I want to be able to show groups these patterns and these effects so that we don't have to wait for the sluggish publication process and writing process in order to start disseminating some of the results.
You referenced that the hippocampus, the volume increases in the hippocampus. What exactly does that mean? Does it, and also, are there particular parts of the hippocampus that you find seem to be more receptive?
That's a very good two-part question. So we found this going back to 2009, the basic answer is we don't really have a good sense on the cellular level right now what's going on. However, what's been interesting is that there's been a number of animal studies that have replicated this effect using imaging. So what they do is they bring animals in, they scan them at baseline, they put them on a running wheel for several weeks, and then they image them again afterwards, and they see changes in the hippocampus, the same types of changes that we see here. Then they're sacrificing the animals, and they're able to look at cellular and molecular changes. So there are changes happening at that cellular and molecular level, but every single study has shown a slightly different pattern right now. So I think that it's still a little bit too early to say exactly what's going on. I would probably say that it can't be just all neurogenesis. I don't think that's realistic. I think that it's probably a combination of a few different things. One is dendritic spine density is probably changing. I would also think that it might be the vascular components, right? So capillary beds, and like I said, animal studies have shown that capillary beds are pretty much a prerequisite for some of the cellular changes. So I would bet that we're picking up some of those capillary beds with the volume of the tissue. And so I think that there's a few components like this, and even increased size of the nucleus as well. So I think all of these things are probably going on, but we're still, I think, a ways away from really being confident there. The second question is about the areas of the hippocampus. So we found in my 2011 paper that the bulk of the association was in the anterior portion of the hippocampus. And that effect also has been replicated a number of times, where it seems to be more in the head and the body of the hippocampus instead of the tail. We have in IGNITE, which I didn't show you here, but we have some data to indicate that there are some subfields of the hippocampus that are more strongly associated with fitness. We haven't yet looked at the intervention effects, but we do think that there's some sub-areas of the hippocampus that are more associated with fitness than other subfields. So for an example, CA2 is not related. CA3 is, and dentate gyrus is, and subiculum is. So we're now getting our measures to at a level of granularity where we can start to pull out these subfields and see these different patterns. And interestingly, this does align with the animal literature as well.
I have a question, Professor Erickson. So I'm curious if there's any research around the different natures of exercise. So to give you an example, some days I'll do yoga, some days I'll play tennis. Mm-hmm. They have the same duration. They have the same level of physical exertion. They both have aerobic part of it, both have a strength part of it. But in playing tennis, it's quite different for the brain. Yep. Because you're playing a match, you're a competition, you have a stress level about that, but you have an arousal, excitement about that. So I'm just curious if that has figured into any of the research, because what I've heard you talk about before was just kind of just aerobic exercise or strength exercise, but without the competitive nature of a lot of games. And of course, one of the major social trends in the United States right now is the growth of pickleball, which I don't quite understand. Yeah. But it's incredible on that. So does getting your exercise that way have some other benefit or some other difference to the way the brain works?
Yeah, it's a very good question. So I'm gonna have my answer be two parts. One is that randomized clinical trials that randomly assign people to receive tennis versus walking, they're not done, right? So we have to rely on epidemiological observational studies, which means that there's people that play tennis, right? We're not training people. There's a select group of people, quite often more socioeconomically advantaged people as well, right? So there's some components there that can influence our interpretations. But when you look at observation epidemiological studies, you can break down effect sizes with risks for cognitive decline and risk for dementia. So there's been a number of these studies that have taken place. One of them actually shows that dancing was the strongest factor. So there's some interesting work going on there. But again, randomized clinical trials in which we randomly assign people to, and you can think of it, walking versus swimming versus tennis versus, like all of these different components, those studies really haven't been conducted. But that being said, we do have some sense. When we look at, for example, yoga. Yoga is a really good example in which we see patterns of yoga on the brain that overlap with a lot of the brain areas that we see with aerobic exercise. Hippocampus is there, anterior cingulate cortex is almost always there in yoga studies. But what's interesting is that yoga also influences the amygdala, whereas aerobic exercise, we never see the amygdala. So there does seem to be some differences in the modality of the behavior that you're engaging in. And you're absolutely right. When people ask me, they say to me, Kirk, I do not like walking around the block or walking on a treadmill. I think it's super boring. I'd rather play tennis. Is that going to have the same effect? Usually my response is, it's probably going to have a greater effect because of exactly the components you mentioned.
So Kirk, I have a question. Your research is truly so elegant, and I think everyone here, I hope, all of us, including me, will exercise more. But I want you to think about exercise is not the silver bullet.
Absolutely.
It's not everything. I want you to talk a little bit about holistic brain health and just how it fits in to a more holistic approach to brain health. And then I have a follow-up question about, since I'll wait for that one, but Advent Health, I'm interested not in pharmacological, but rather benefits. Would you go to an insurer to say, what if I could save you money by, if you paid your clients to exercise, have you gone that route? But I want you to talk, first of all, to put exercise in more of a holistic approach to brain health.
Absolutely. We all recognize that exercise doesn't happen in a vacuum outside of all of our other behaviors, sleeping, eating, and I mentioned, going for a run and stopping for a greasy burger. Having a greasy burger now and then is perfectly fine, but if you do that regularly, of course, it's going to be impacting any benefits you're getting from going for a run. So these things all work together. Right now, we're studying physical activity in isolation from dietary behaviors. In Ignite, we're not manipulating diet, but we measured it. For the first time in a big study like this, we measured it. So we're now able to examine things like a healthy eating index or a glycemic index based on the dietary patterns that somebody has and examine how this is influencing the outcomes and measurements that we have collected. So I do think that we have to be thinking holistically about our entire daily activity, our daily routines. One really interesting finding that I was just mentioning at the table over lunch here is that there's analytical approaches to now examine the composition of your day. So this is called a compositional data analysis, a CODA approach. You can think of it as in terms of like a body composition. Right? So we have our composition of our body. We can determine the percent fat, for an example, in our body. And so when you think about our 24-hour day, there's no more than 24 hours, unfortunately, right? So we're stuck at 24 hours. So how do we think about the composition of that day, and can we determine the best recipe for maximizing brain health? From balancing the amount of sleep versus the amount of sitting versus the amount of physical activity that you're getting. And so we and others are starting to apply this approach. And one of the recent findings that we have is that engaging in moderate to vigorous physical activity was the driving force around the associations with cognition. So what's interesting is that if you take only 10 minutes away from sleep and you put it towards physical activity, that's a bigger impact than putting 10 minutes into sleep. Okay? So these types of balancing is, I think, what's really important and what's going to be really important for future research to be starting to untangle. In addition, I can't remember what your other...
My other question was about insurers.
Oh, yeah, insurance. One other thing before that. There's some really interesting research on exercise and cognitive stimulation. That exercise is a great way of creating the foundation, but unless we really use our brains, we're not benefiting as much as we would like, right? So using your brains is critical when you're growing them. And so this is a critical piece. Now, unfortunately, there's actually not been a whole lot of studies that have tried to put these together, actually. And so that's an area where we need more research as well. In terms of the insurance, well, it's hard for me to share my views on insurance companies in a public gathering. I don't think very highly of insurance companies, let's say that. And so, yeah.
I exercise every day, and I was exercising next to a young woman who, she was wearing a Fitbit, and she said that her company paid for her insurance if she exercised regularly.
Yeah.
And
Yeah.
she got and they got a discount on it.
Right.
So I think, to your point,
There are companies that are doing this, offering incentives. There are some insurance companies that are doing this as well, and they're monitoring it. They're monitoring your gym membership and how often you go and your activity levels. So there are some insurance companies and companies that are recognizing the importance of it. Not enough, in my opinion. Not enough that even recognize the importance of prevention, period. So it's a constant struggle, but I think we're starting to see strides slowly, but yeah, it's like moving heaven and earth sometimes to get anything done with insurance companies.
That seems a fairly down note to end on, but we are at time. I'll apologize to our online audience. They were tremendous in terms of their questions. Fortunately, many of them overlapped with what you did talk about. So thanks so much for joining us online, and thank you for being here. And let's again thank Dr. Erickson for this amazing presentation.
Thank you so much.